Thank you, everybody, for joining us today. So as she said, this is more of a focus on managing larger mouse colonies and the use of ovarian transplants. And I'm going to give you some examples of how JAX has used them, as well as kind of the process and the different things that we look at in using ovarian transplants. Oh, my apologies. So using the ovarian transplants, there's many reasons to utilize ovarian transplant process and can give you great benefits in regards to colony management of mouse strains. The things that we've seen that have been benefits for us is we've increased preferred genotype ratio of offspring, which in turn allows us to reduce our total colony sizes needed to produce what we need for our scientific community. We've been able to increase longevity of breeding by using ovaries from a strain that has a morbid phenotype into a host that does not. We've eliminated genome typing costs, and a lot of these benefits do revolve around increasing our margin and being able to provide more animals with less resources. We've been able to improve one of those 3 Rs by reducing animals born of undesired genotype, and then utilizing ovaries from a mouse that normally would not be able to naturally produce due to their phenotype. So this is just a basic diagram of how the ovarian transplant process works for us. You select a female with a desired genotype that you want to mate. You remove their 2 ovaries, cut each of those ovaries in half, and then you transplant them into 4 different hosts or recipients. So you get a 1-to-4 ratio, 1 female to 4 recipients for mating. Then you take a male and you mate that to those hosts, and the offspring of those will then be generated from the mating, either the genotypes you're looking for or the phenotype. There are issues that you can have with the ovarian transplant process in which you can have an incomplete OT. And when that happens, you would produce mice that are of the original coat color of the host. And that just tells you there's 2 different things that can happen. Either you have a partially incomplete OT, in which some mice within the litter will have the same coat color as your host, or you may have an entire litter that is the same coat color as your host. And that just tells you that either the host, the ovarian transplant did not work, and so it's producing all mice from the host female, or the ovary of the host female is still partially working, and you get offspring that represents the host mouse. So something we have to really consider is host considerations when selecting to use an ovarian transplant process. The first thing is you want to make sure that they have compatible haplotypes between host and the donor, and that's key so that rejection of ovaries does not occur. And then, as I pointed out in the last slide, the coat color of host versus recipient. In the first example, if your donor has black fur and your host also has black fur, all pups that are born will also have black fur, and you would not be able to identify if incomplete OT was done. To be able to manage that so you don't have the option of having a host that is of a different coat color, you would have to genotype all of your offspring to be able to identify the segregating genotypes. So in our cases, we like to choose a host that has a different coat color so that if incomplete OT does occur, then pups that are the same color as the host, you know, are from the host mouse, and the ones that are the same coat color as the donor are of the correct strain. More host considerations that we like to think about is really the host strain and their productivity. You wouldn't want to choose a host strain that has, small litters or are not very good mothers. So we look at different aspects of the host strain itself and make sure that all of those qualities are ones that we would want to have with a successful ovarian transplant to be able to breed them through a long breeding life. Another thing is adequate weight at wean age. We like to do the ovarian transplants at a young age due to it's less bleeding that happens during the ovarian transplant process. So we want to make sure we have robust mice at wean age for the surgeries. You also want to make sure that there aren't any known phenotypic or genetic variables that would limit the mother's ability to nurture or produce pups after ovarian transplant. And the really key thing that we do with anytime we're considering converting a strain to ovarian transplant is really trial and error, and trial and collecting data and making sure that the host that you're choosing is appropriate. It could look good on paper. However, in one of our examples, I believe we went through 7 different host strains or combinations of strains to create a hybrid until we found the one that worked best for that strain. So now I'm just going to go through 3 different examples that we've used ovarian transplants for and the benefits we gained from it. The first one is the JNU strain, which is our outbred nude colony, and it's historically made heterozygous females by homozygous males, which should, in the Mendelian world, give you about 50% genotype ratio of each. For this strain, the homozygous mouse is our preferred product, and so we should be getting about 50% of that product. But because this strain is outbred, they produce very large litters, which are between 10 and 12. And the larger litter sizes do have an effect on the survivability of the home. They're more fragile than the heterozygous, they're nude. So what we did in the colony management is to put in a process that we would pre-wean out prior to wean age heterozygous to reduce the litter size to be able to increase the survival rates of the homozygous mice. Even with that practice, the genotype ratio didn't reach the expected results, and we were seeing about 40% hets and 40% homes and 60% hets. Because of this decrease in genotype ratio, we have to have more breeders on the shelf to be able to hit our goals. So you would think to yourself, well, why not just breed home female by home male? That would solve all of your problems. Well, we tried that, and we found that the homozygous females do not breed well, and they tend to struggle with nurturing their litters. So we used the homozygous ovaries and transplanted them into an immune-deficient host and mated them back to a homozygous male from the colony. And by doing this, we had 3 big key benefits from it. The biggest one is the preferred genotype ratio went from 40% to 100%. This equated to approximately a 60% reduction in breeder boxes required to produce our target. That's huge as far as colony management is concerned. We also eliminated the need to pre-wean heterozygotes out at 2 weeks of age to increase survivability and reduce the production of non-preferred. So this is more of like producing mice that you're ultimately going to discard because you don't need them. So that was something that was very key to my heart is trying to reduce the number of mice that we were making that we were not going to be using. So utilizing ovarian transplants allowed us to eliminate this process. And the other thing that we found really interesting is that due to smaller litter sizes, which you would think would be a bad thing, we actually saw a decrease in the pre-wean loss, which reduced and resulted in more animals making it to wean age. And Transplant 4 is our OB-OB and DB-DB models. These are models that carry a spontaneous mutation that causes obesity and diabetes. Due to these phenotypes, the homozygous females and males are not able to produce naturally, but the ovaries of the homozygous females are completely functional. So I'm going to try to get my pointer here. There we go. So we utilize homozygous female ovaries. So if we look at, this is our historical mating here. Before OTs, we would use heterozygous females by heterozygous males, and that would result in a 25% of our preferred product, which is the OB-OB, 50% of the heterozygous, and then a 25% of wild types. But by utilizing the homozygous female ovaries, here we were able to increase our preferred genotype ratio to 50%. And then we're also able, which is key, to eliminate this genotype here. And what that also allowed for was that in our historical mating, if the mouse was not obese, then we would have to genotype those mice to be able to identify if it was a het that we could use back into mating or a wild type that we would discard. With changing to ovarian transplants, we were able to eliminate that genotyping cost, eliminate that genotype altogether, and be able to just identify with looking at the mice, the phenotype that we had our OBs and then we had our hets. And then also for utilizing this, if we were ever needed to produce obligate hets, we could utilize ovarian transplants to mate back to the inbred and produce all obligate hets for breeding. The last example I'm going to go over is our Huntington's model in ovarian transplants. The Huntington's mouse model have a severely shortened lifespan due to disease onset, and it poses a problem when we try to breed them because of the very short lifespan. It's not efficient, and we also have to keep animal welfare in mind. For this transplant process that we did, because we have always utilized ovarian transplants in a certain fashion, but it wasn't, in this case, wasn't about increasing genotype ratio. Rather, it was to increase productivity and not have to use hemizygous males that would eventually show a phenotype. So because of this disease, it results in infertility and shortened breeding length. Ovarian transplants with hemizygous females to a hybrid host allow for a longer breeding rotation in which the hosts do not succumb to disease onset and therefore can produce more litter throughout the rotation. The other thing we have to keep in mind in this strain is the CAG or triplet repeat of these strains plays a part in the disease in the breeding of the colony. Besides, the CAG repeat region directly affects the age of phenotype development. And through our own experience, it was assumed that the CAG repeat number for this strain decreases 1 to 3 repeats in weaned from OT hemi female by hybrid male mating, and that the CAG number increases 2 to 3 repeats from wean in a hybrid by hemi male mating. So Jack's developed a breeding scheme in which it utilized wean from both matings to kind of try to get a stable CAG range amongst all of the offspring. So to prove or disprove this theory, a trial was done using ovarian transplant hemizygous females mated with hybrid males for multiple generations, and the results were quite remarkable. And I don't think a lot of us, I know Andy, who's on this panel, saw the results as well, and it was something we had never seen before or been able to get data for, so it was really quite amazing. But the study was trial was to study 2 different aspects, and really what happens when you breed through the female by ovarian transplant over several generations. Does it prove or disprove the theory that it increases or decreases if you breed through one or the other? And are the ovaries affected by the disease, and so is CAG repeats affected as the OT recipient and ovaries age? We used 2 different Huntington's models for this trial, one that had a lower CAG repeat range, which results in delayed disease onset, and one that has a higher range, resulting in earlier disease onset. So the next 2 slides are just going to show you the data that we collected from both of our models. The first one is one that has a lower CAG range. And what's really interesting to see is that, so what we did was we took mice from the first generation of OT matings and then took wean from that and mated second, and then took wean from second to third to fourth, and mapped out where the CAG numbers fell within a given range. And as you can see, the CAG numbers, as the ovaries age, start to trend down. And they start to trend down at or around the same point at every generation. Now, for our own purposes, we can use this data to decide, okay, yes, the CAG number does decrease, but at what point? And at what point are we able to extend our rotation or look at, the productivity of the mice as the ovaries age? And then this is slides for our colony that has a higher CAG number. And this one, they start to shift sometimes a little bit sooner than the other strains, but they follow the same pattern, which is really interesting to see, that CAG number is affected by the age of the ovary of the donor female. The more conclusions we found through this trial was that, in our opinions, there is success in using OTs over several generations. Each generation, the data was very close to the previous generation. We did find that CAG repeats trended down. We noticed that we can increase our rotation length thanks on our current processes. We could actually extend it several weeks beyond what we're currently doing. The productivity didn't change as the ovaries aged. Litter sizes stayed the same. The pre-wean loss stayed the same. The only thing that really changed was that CAG range average. And then the mating scheme change. This allowed us to be able to just breed through one mating scheme. We would not have to utilize male hemizygotes that are going to succumb to this disease for breeding and keep a consistent breeding scheme throughout. So in conclusion, basically there are lots of benefits to using ovarian transplants, most of them listed here, but a good thing to point out is that prior to starting anything, data collection of host strains, productivity is crucial in identifying the host to get the best results. We have had instances where things go very well. We've chosen a host and they've bred well, and then we find over time that the phenotype of the mice that are being produced are not what our main product is, and we don't know if that's epigenetics. We don't really know exactly what is the cause of it, but there are some cases in which utilizing ovarian transplants may not be beneficial. So I want to say thank you for everybody for coming and listening.